Hello, welcome to my channel. So today we're going to go through cells. So we're going to go through the human cell to be specific. Okay, so let's start. So in terms of introduction, the cell from the Latin word cellula means a small room and is a basic structural and functional unit of life forms. And every cell consists of a cytoplasm enclosed within a membrane, which contains many biomolecules such as proteins and nucleic acids. So initially it was discovered by Robert Hooke in 1665, and the cell has a rich and interesting history that has ultimately given way to many of today's scientific advancements. Then the first cell is thought to have arisen by the enclosure of self-replicating RNA and associated molecules in a membrane composed of phospholipids. Then each phospholipid molecule has two long hydrophobic. Then by the late 1830s, botanist Matthias Schleiden and zoologist Theodor Schwann were studying tissues and proposed the unified cell theory. And the unified cell theory states that all living cells are composed of one or more cells. Then the cell is the basic unit of life, and new cells arise from the existing cells. That was the introductory part, and this is the section review of a human cell. Okay. Then cells are composed of water, inorganic ions, carbon containing organelles. Then water is the most abundant molecule in cells, accounting for 70% or more of total cell mass. Consequently, the interactions between water and the other constituents of cells are of central importance in biological chemistry. Okay, so this is about cell diversity, different types of cells, which you mentioned in your previous introduction in anatomy and physiology. Okay, so these are the cell diversity: nerve cells, fat cell, epithelial cells, and whatnot. Okay, then parts of a cell. So the human cell are three main parts. We have the plasma membrane, which is the outer boundary of the cell, and they have the cytoplasm, which is the intracellular fluid packed with organelles, which is the small structures that perform structure of cell functions. Then you have the nucleus, which is an organelle that controls cellular parts. The nucleus lies near the cell's center. Okay, these are the main parts of the cell. Okay, so we talked of the cell membrane, the nucleus, and the cytoplasm. That's the main parts of a animal cell, which is specifically we are going to talk about the human cell here. Okay, then the plasma membrane defines the extent of a cell, thereby separating two of the intracellular fluid within cells and the body major fluid compartments, the extracellular fluid outside cells, of course. Then the plasma membrane encloses cell contents, mediates exchanges with the extracellular environment, and plays a role in cellular communication. Then it is selectively permeable plasma membrane that permits some substances to pass more readily than others. Then a concentration gradient is a difference in the concentration of a chemical from one place to another, such as the inside of the cell to the outside of the plasma, from the inside to the outside of the plasma membrane. Okay, so this is the plasma membrane we're talking about. Okay, and this is how to draw the plasma membrane and its cell structure. So you have the phospholipid bilayer, you have the ion channel protein, the glycolipid, the peripheral protein, the integral protein, glycoprotein, and cholesterol. In terms of structure, we have what we call the fluid mosaic model, where the fluid mosaic model of membrane structure depicts the plasma as an exceedingly thin, about 7 to 10 nanometers, structure composed of double layer of lipid molecules with protein molecules dispensed in it. Then the proteins which float in the fluid lipid bilayer form a constantly changing mosaic pattern. Okay, then the lipid bilayer, the basic structural framework of the plasma membrane is a lipid bilayer, which is a two back-to-back layers which is made up of three types of lipid molecules: phospholipids, cholesterol, and glycolipids. About 75% of the membrane lipids are phospholipids. Lipids that contain phosphorus, present in small amounts, are cholesterol, about 20%, and a steroid with attached with an attached hydroxyl group, and various glycolipids, which is about 5%. Then lipids with attached carbohydrates groups are also present. Then the bilayer arrangements occurs because the lipids are amphipathic molecules, which means that they have both polar and non-polar parts. So in phospholipids, the polar part is the phosphate containing, which is the head, which is hydrophilic, or meaning hydrophilic means water loving. Then the non-polar parts, which are the two fat acids, which are the tails, are hydrophobic, meaning water fearing, and hydrophobic hydrocarbon chains, because as they say, like seems like seeks like, so meaning they attract each other. The hydrophobic parts are going to attract each other. Okay, so the phospholipid molecules orient themselves in the bilayer with their hydrophilic heads facing outwards. Okay, and in this way, the heads face a rotary level on either side, and which is the cytosol on the inside and the extracellular fluid on the outside, and the hydrophobic fat acid tails in each half of the bilayer points toward one or another, forming a non-polar hydrophobic region in the membrane's interior. So that's the fluid mosaic model. Okay, and this is the phospholipid bilayer. As you can see, this is the water part and the water part. So meaning the ones which are facing the water are the hydrophilic heads, which are water loving, and the ones which are away from the water are the hydrophobic tails, which are water fearing. So together, this is the makeup of the phospholipid bilayer. Okay, hope diagram is clear enough. Okay, so moving on. So there are some structures within the structure of the plasma membrane. We have what we call the glycocalyx, which is where the plasma membrane contains a carbohydrate-rich area at the surface, at the cell surface. We call the glycocalyx. Then that's all in the plasma membrane. Then we can talk about the cytoplasm, where the cellular material between the plasma membrane and the nucleus, which is the site of most cellular activity. And it consists of three elements. You have the cytosol, you have the organelles, and we have the inclusions. So in terms of the cellular material between, in terms of the site, describing what the cytosol is. So the cytosol is the viscous semi-transparent fluid in which the other cytoplasmic elements are suspended. Then the organelles are the metabolic machinery of the cell. So each type of organelle carries out a specific function for the cell. Then inclusions are the chemical substances that may or may not be present depending on cell type. Okay, then going through the organelles. So these are the organelles. Now we are going to go through the organelles. So starting with the mitochondria, as shown by the structure, it is a rod-like, rod-like double membrane structures, and the inner membrane looks folded into projections called cristae. Okay, this is the folded projections part. Then it is the site of ATP synthesis, known as the powerhouse of the cell. Okay, then the other organelle is the ribosome. So ribosome is a dense particles consisting of two subunits, each composed of ribosomal protein and RNA, free or attached to rough endoplasmic reticulum. So this is the ribosome, and we have the large unit and small subunit, which together make up the ribosome. Then they act at the site of protein synthesis. Okay, then we have the rough endoplasmic reticulum. So this is a membranous system enclosing a cavity where we have the cisterns and the coiling through the cytoplasm, externally studded with ribosomes. So we call it rough because it has ribosomes on its surface. Then sugar groups are attached to proteins within the cisterns, and proteins are bound in vesicles for transport to the Golgi apparatus and other sites. The external surface synthesizes phospholipids. Then we have the smooth one, which doesn't have ribosomes on its surface, which is a membranous system of sacs and tubules free of ribosomes. It is the site of lipid and steroid, which is a cholesterol synthesis, lipid metabolism, and drug detoxification. That's the function. Then you have the Golgi apparatus, which is a stack of flattened membranes and associated vesicle close to the nucleus. And it is packages, modifies, and segregates proteins for secretion from the cell, inclusion in lysosome, and incorporation into the plasma membrane. And this is the structure. Then peroxisomes are membranous sacs of oxidase and catalase enzymes. Okay, and the enzymes detoxify a number of toxic substances, and the most important enzyme, catalase, breaks down hydrogen peroxide. Okay, then we have lysosomes, which are membranous sacs containing acid hydrolase, and act as sites of intracellular digestion, and this is their structure. And lysosomes are involved with various cell processes, and they break down excess or worn out cell parts. They may be used to destroy invading viruses and bacteria. Okay, then we have microtubules. Okay, so for microtubules, this is the structure of the microtubules. Cylindrical structures made of tubular proteins, and they support the cell and give it shape, and they are involved in intracellular and cellular movement, and they form centrioles and cilia and flagella if present. Okay, then in terms of microfilaments, so microfilaments, these are the structures for the microfilaments. So microfilaments are fine filaments composed of the protein actin, and they are involved in muscle contraction and other type of intracellular movement, and help form the cell's cytoplasm. So I think the cytoplasm part was obstructed. Okay, so that's about the microfilament. So we have also what we call intermediate filaments. So intermediate filaments are protein fibers. Composition varies. Then the stable cytoskeleton elements resist mechanical forces acting on the cell. That's the function. Then we have centrioles, and this is the structure of a centriole, which are paired cylindrical bodies, each composed of 9 triplets of microtubules, and they organize a microtubule network during mitosis, which is cell division, to form the spindle and asters, and form the bases of cilia and flagella. Okay, then you have inclusions. Inclusions include stored nutrients such as lipid droplets and glycogen granules, protein crystals, pigment granules. Then storage for nutrients, wastes, and cell products. That's the function of the inclusions. Then the cellular extensions. So the major cellular extensions are cilia, flagella, and microvilli. Okay, so in terms of the cilia, this is the structure of the cilia. This is the structure of the cilia. These are short cell surface projections. Then each cilium is composed of 9 pairs of microtubules surrounding a central part. Then coordinated movement of cilia creates a unidirectional current that propels substances across the cell surfaces. So that's the cilia, and this is the epithelial tissue. Okay, then the flagellum, like a cilium but longer. Only example in humans is the sperm tail, which propels the cell. That's the sperm cell, and this is the flagella here. Okay, and this is the structure of the flagella. Then the microvilli, these are tubular extensions of the plasma membrane. It contains a bundle of actin filaments and increased surface area for absorption. Okay, then in terms of the nucleus, we can talk about the nucleus. So the nucleus is a spherical or oval-shaped structure that usually is the most prominent feature of the cell, and it is surrounded by the nuclear envelope and contains fluid, neoplasm, nuclei, and chromatin. Then it is a function is act as a control center of the cell, responsible for transmitting genetic information and providing the instructions for protein synthesis. So in terms of the structure of the nucleus, we have the nuclear envelope, the neoplasm, the chromatin, and the nucleolus. And this is the structure in terms of the figure. Okay, we talked of the nuclear envelope, the nuclear pore, chromatin, nucleolus, and it's the surrounding part is the endoplasmic reticulum. But in terms of the structure, these ones which you have highlighted are the anatomy of the nucleus. Okay, then going further in terms of the nuclear envelope. So the nuclear envelope is a double-layered membrane perforated with pores, which controls the flow of the material going in and out of the nucleus. Then you have the outer layer, which is connected to the endoplasmic reticulum, communicating with the cytoplasm of the cell. Then the exchange of the large molecules, such as protein and RNA, between the nucleus and the cytoplasm happens in the nuclear envelope. Then the neoplasm is a jelly-like, made mostly of water, matrix within the nucleus. Then all other materials float inside the same nucleus. Then it helps the nucleus keep its shape and serve as the transportation media, serves as for the transportation the median of important molecules within the molecules. That's the function of the neoplasm. Then finally talk about the chromatin. So chromatin appears as a fine and evenly stained network, but special techniques review it as a system of bumpy threads weaving through the neoplasm. Then chromatin is composed of approximately 30% DNA, which is our genetic material, then 60% globular histone proteins, which package and regulate the DNA. Then 10% RNA chains, newly formed or forming. Then chromosomes contain DNA in a condensed form attached to a histone protein. Then chromatin is comprised of DNA, and there are two types based on the function. We have what we call euchromatin, which is a less condensed and can be transcribed. Then the second form of the second form we have what we call heterochromatin, which is highly condensed and typically not transcribed. Then under the microscope, in its extended form, chromatin looks like beads on a string. Then functions, the nucleus is often compared to the command center, as it controls all functions of the cell, and it is important in regulating the actions of the cell, and it plays an important part in creating the cell's protein. It is also involved in important processes dealing with the DNA and other genetic materials. Okay, then when we talk about the DNA itself, so the DNA means deoxyribonucleic acid, which contains the information needed for the creation of proteins, which includes enzymes and hormones, and is stored in the nucleus, as already said, and then in the form of chromatin or chromosomes. Then the nucleus is the site of DNA duplication, which is needed for cell division in mitosis and organism reproduction and growth. That's the DNA structure. Okay, so then in terms of RNA stands for ribonucleic acid, which are made from the DNA template. We have different types of RNA. We have messenger RNA, which directs the synthesis of a protein. We have ribosomal RNA, which joins the ribosomal proteins to make ribosomes. Then we have the transfer RNA, which binds to an amino acid and holds it in place on a ribosome until it is incorporated into a protein during translation. Okay, then in terms of proteins and cell regulation, the nucleus oversees functions and regulatory mechanisms for keeping the cell healthy and alive. Then the nucleus also controls growth of the cell through the synthesis of structural proteins, energy, and nutrient metabolism. Then the nucleus also regulates the secretion of ribosomes, which are made in the nucleolus and are the sites of gene transcription. So that's the end of about cells. So next we can talk about movement of particles inside the cells as another tutorial video on the anatomy and physiology section. Okay, so that's the end of this one, and if you haven't subscribed, please subscribe and hit the bell notification, and check out the anatomy and physiology playlist so that you actually have ideas on the combined anatomy and physiology slides. And thank you for watching once again. Please subscribe and hit the bell notification. See you on the next tutorial.